pill for acute spinal cord injury, if there was a pill for once and for all to cure Parkinson's disease or ALS, Lou Gehrig's disease, we wouldn't have to be working on this. We're not doing science for science. We're doing science to benefit patients. And then also, I think it's most important to manage the expectations of our patients with responsibility. That's why I gave you those data about clinicaltrials.gov being the home of where FDA-approved clinical trials are being executed on adult stem cells. The first filing for human embryonic stem cells for acute spinal cord injury, as I told you the story, was paused on May 13th, 2008. It's imperative. What we can tell you is that, yes, we can generate human brain cells that behave like their in vivo counterparts. Yes, in some animal models of disease, when we transplant those cells, we're able to revert what we call the phenotype or the symptoms. But we have to manage your expectations with great responsibility. And also, just think about it for a minute, because we hear, oh, where's the cure? Where are the therapies? When are they coming out? It takes 15 years to get a drug to the market. 15 years. In a process that we know, the drug industry has been around since how long? Human embryonic stem cells are only 10 years old. Right? So now, while we're learning how to differentiate, how to make these therapeutic cell types, so it's a request, we always have to be very aware. This is a new era, no question, that these cells might serve as a vehicle for a new area in medicine, the era of regenerative medicine. But we also have to be realistic. It takes 15 years to get a drug to the market in an industry that's highly developed that we have been working on for hundreds and hundreds of years. So ultimately, and this is my last slide, I want to, the take-home message is that stem cell research benefits people at so many different levels. First and foremost, as we think and who we serve are our patients. So we are striving through innovation to address your unmet medical needs. But it also benefits the community. Madison is one example. It attracts investment dollars from the East Coast, from the West Coast, to the Madison community. It grows the community. It creates jobs in the community. It benefits us in being able to contribute to medical advances and really make a difference. I mean, the work that I'm doing with autism is such an interesting story because we don't understand the causes. The model that I've developed is it's a model. For example, the minority of the children with autism have autism due to valproate exposure, not the majority. But even if it's a model that can help us to understand the causes of autism, even in the minority of kids, that we hope that will be able to make a difference. And then last and not least, it's really an opportunity to develop the people of Wisconsin and the people in Wisconsin. I have so many of my students that grew up here, that continue to live in rural areas of Wisconsin, and they have the opportunity to travel the world and present to the world about their human embryonic stem cell research. So thank you very much. This is my lab. This is Jessica Quam, who was my first graduate student, and now she is my laboratory manager. So I'll take any questions. Thank you so much. Go ahead.
How do you, in layman's terms, how do you differentiate the use of human embryonic stem cells and cloning?
There's no cloning. Simply put, there's 150 or more people on this campus working on human embryonic stem cells. None of us work in cloning.
But what's the difference?
So to clone, actually, you would have to, how do we clone an animal? We get a skin cell from, let's put it, we did this with cows and we've published. You get an ear notch from a cow and you inject that into an embryo of another cow, and then you create an embryo that's cloned so you take the DNA out of the embryo from the other cow, and then the DNA now comes from that skin cell that you injected, and that's how you make a cloned embryo. Now, when that cloned embryo develops, it develops in the same way as, let's go back. Remember that embryo that I showed you, the picture in the beginning? So a cloned embryo develops in the same way as a normal embryo. And a cloned embryo would have embryonic stem cells to be derived. Does that make sense? So you could create embryonic stem cells through cloning.
Right.
But you don't, but none of us, I mean, that's really, it's been one of our biggest challenges, and we really don't know where the problem is started that we got associated with cloning. I'll tell you when it started. It started with the possibility of, okay, so you have these human embryonic stem cells that came from embryos that have a different genetic match. Say I'm a Parkinson's patient and I would like brain cells with my genetic match. So potentially I could clone myself to generate brain cells that have a genetic match to me to transplant back to me, right? So that's theoretically, that's where somatic cell nuclear transfer, which is cloning, comes into play in the big picture of things. Now, with Jamie's breakthrough discovery of the iPS cells, there is if theoretically we wanted to create a brain, I'm a Parkinson patient with Parkinson's disease. I want brain cells with my DNA. Jamie's breakthrough discovery of creating these induced pluripotent stem cells shows that we could potentially take a piece of my skin, reprogram them back in the lab, nothing to do with cloning. We change their DNA in the lab, nothing to do with cloning, and then those cells could make brain cells for transplantation.
Well, why can't we use the clone cells?
Why?
Seems a lot easier.
None of us work in cloning. None of us work in cloning. So I'm the one to speak because I did my PhD in animal cloning. So for animal cloning, there was a reason. Why were we cloning animals? We were cloning cows in DeForest, Wisconsin, because these cows had a protein in their milk for hemophilia, patients that bleed to death, that need to get blood transfusions. So there was a reason for cloning. To propagate endangered species. We tried to clone a gar. It was born and it died after birth. I wasn't involved directly in that project. But for regenerative medicine, there's no need whatsoever. At all. Go ahead.
A lot of countries do not have the restrictions that the FDA puts on for developing medicines. How do you anticipate that as things develop, will we be kind of...
We are already, my global view of things, and I do travel a lot, in my global view of things, we are already lagging behind. The UK, for example, has leading and effective policies. The UK has not spent so much time, running debates and immediately saw the opportunity, and it has created these policies light years ahead of us. And when you go to the UK or even in Canada, I was in Canada the other day and people ask me, what do? You're the waitress at the restaurant. I go, I do stem cell research, human embryonic stem cell research. Why are you whispering? That's the attitude in Canada and the UK is, so even in terms of public perception, we know that the American public supports stem cell research. As Bernie mentioned, these polls have shown 70–75% of people. But in practical manners, which is your question, yes, we are lagging behind. So it's likely that we will see therapies, being developed in a faster way in other countries. But we're on the right track. The fact that the FDA has